off of the main path of the alimentary canal and talk about the pancreas and the liver, accessory organs of digestion. Required, but not part of the path where food and chyme go. So we're gonna, well, we'll start with the stomach and we'll talk about gastric ulcers because it's a pretty interesting story, some modern pharmacology. Then we will talk about the pancreas, then we will talk about control of the pancreas, and we'll start to unravel some of the complicated anatomy of the liver. That's where we're going today. Let's start with a clicker. Let's just activate, build up a little bit. Gastric acid is answer is B and D. Parietal cells are the cells that secrete acid, and the acid is hydrochloric acid. Okay, gastric acid secretion is stimulated by maybe it's somatostatin inhibits the secretion of gastric acid. So let's just talk for a minute about ulcers. You've heard about this. You've probably seen the ads for various ulcer medications. Ulcers are holes, wounds either in the intestinal wall or in the stomach wall, and they're painful. They can be dangerous because of excessive bleeding. And so they are treated pretty aggressively, both gastric ulcers in the stomach and intestinal ulcers, primarily in the duodenum. The drugs that you've seen advertised in magazines or TV fall into two main categories. Tagamet and Pepcid and Zantac, you've seen these ads. These drugs are histamine blockers. They inhibit they block the histamine receptor. And they're specific, so you may have heard of antihistamine drugs that you take for things like, sinus congestion. Those are non-specific antihistamine drugs, non-specific histamine blockers. These drugs are specific for the histamine receptor that is expressed on parietal cells. So they're not what we call, so they don't have an antihistamine like, for sinus and for nasal congestion, the way something like, what's the one I'm thinking of? Antihistamine. The one that's in the pink pills. Benadryl. Benadryl is non-specific. These drugs are specific for this histamine receptor. And so they decrease acid production, and they decrease the tendency for acid to, hydrolyze mucus and cause damage to the stomach or the duodenal wall. The second class of drugs, again, you've seen these, Prilosec, Nexium, the purple ones. They are blockers. They are blockers of the pump itself. So they don't block a receptor. They are proton potassium pump blockers. They inhibit the pump. And again, to inhibit acid production. The idea is that ulcers are caused by excess acid. And so some people take antacids, like Tums. Tums are drugs that you take to buffer the acid. So they don't do it. They chemically reduce the amount of acid, and they can be effective, but not as effective as these drugs. But also not as expensive as these drugs. We can get into the politics of drug discovery and drug pricing and insurance. That's a thing, but we're not going to do that. And there's a really interesting story about ulcers that is quite a few years old now, but I think it's still kind of inspirational. So these two guys they're Australian gastroenterologists, and they had patients who had ulcers, And they had this idea and some evidence that ulcers were not just caused by excess acid, but were also caused by excess proliferation of a bacterium, Helicobacter pylori, a bacterium that thrives in the acidic environment of the stomach and the early duodenum. And the idea, we all have this bacterium in our stomach, but it's kept under control by useful, biomic interactions with other bacteria and with our environment. But some people, this bacterium can proliferate, and they were convinced that this was a contributing factor, and maybe the contributing factor, to the start of a gastric or duodenal ulcer. And so they had some evidence, and they published it. They tried to get their paper published in a we're not talking about Science and Nature here. We're talking about the journal. They couldn't even get their paper published in the annual meeting of the Gastroenterological Society of Australia. This is a specialized, I'm not making fun of Australia. This would have happened in the US too. There's a specialized audience of gastroenterologists had ruled, and some committee had ruled, nah, this can't be it. It's acid. It has to be acid. There's always been acid, it will be acid, it's acid. Helicobacter, we're not even going to pretend to entertain the possibility that it's Helicobacter. And so it was rejected. So it was rejected in March of 1983. And in October they persisted. October of 2005, they won the Nobel Prize for the discovery of Helicobacter's contribution to gastric and duodenal ulcers. So what is that? How many years? 83, 93, 2003, 23 years of persistence. Obviously they didn't, so they were gaining traction over 23 years, and 23 years between the rejection from the Gastroenterological Society of Australia to the awarding of the Nobel Prize. So I think for a scientist there can be no greater validation of the value of persistence than this story, right? They could have just said, oh, okay, fine, whatever, then nobody believes it, we give up. And they didn't. And they won the Nobel Prize for it. So here you are, stressing about your exams, holding on for dear life to make it to the end of the day on Friday. Persistence counts. Thank you. Okay, so now let's talk about the pancreas. And we're specifically going to talk about the exocrine pancreas, not the endocrine pancreas. The pancreas has two main functions. The endocrine pancreas is to secrete things like insulin and glucagon for the control of blood sugar. And we talk about the endocrine pancreas in AMP1. Now we're going to talk about the exocrine pancreas, the pancreas that is required for digestion. So the exocrine pancreas, just like the exocrine salivary glands, there are acinar cells at the end of a duct and duct cells that line the duct. And these ducts the duct goes from the pancreas into the duodenum. So this is secreting digestive juice from the pancreas into the duodenum for the digestion of proteins, fats, and carbohydrates. Well, not so much fat, but yeah, a little bit of fat. All three of the main food groups require pancreatic exocrine secretions. So the idea is that the acinar cells at the sort of cul-de-sac or dead end of these ducts does a lot of the secreting, but the duct cells that line the ducts also secrete. They're both secretory organs. One is not, it's not just plumbing. So the enzymes that the acinar cells at the end mostly secrete enzymes. And the enzymes that they secrete are listed here. We'll go through them in some detail. Amylase that you've heard about from saliva for the digestion of starch. Lipase that you heard about from lingual lipase in saliva that is important for the digestion of fat. And these three ogens, zymogens, these are precursors. Well, this one is clearly a precursor because it's got the ogen. This one's is the ogen, it's a precursor. And this one actually should be pro-carboxypeptidase, which the pro indicates that it's a precursor as well. These enzymes are important for the digestion of protein. These are peptidases. And so we'll talk about these enzymes one at a time, and then we will also talk about why we need to secrete, the triggers for bicarbonate. Why we need to secrete why our pancreas needs to secrete bicarbonate should be pretty obvious. There's acid moving from the stomach into the duodenum. We don't want that. The bicarbonate neutralizes gastric acid. So here is a pancreatic acinar cell. So this is an actual image of one of these cartoon cells here, right? So there's the acinus, or dead end of a duct. So we're zooming in on one of these cells, and in reality, not cartoon, this is the way they look. So here's the lumen of the pancreatic duct. Here is the cell with the nucleus and the rough endoplasmic reticulum, and it is chock full of rough ER because what we have is a robust collection of vesicles that are chock full of those enzymes, or proenzymes, or ogen enzymes, that are getting ready to be secreted into this lumen and flowing down the duct to the duodenum. So these cells are among the most they have the most proliferative and rough endoplasmic reticulum. And you can remember some things from cell phys about how the rough ER is necessary for the production of proteins that end up in secretory vesicles. Remember the co-translational translocation of proteins, right? Ribosomes docked on the rough ER, translate the RNA for these proteins through the ER membrane into the ER where it is processed, and then we have the Golgi, and then we have the secretory vesicles. I hope I'm triggering some memory of a pathway that you learned about in cell phys. I'm not going to test you on it, but I'm hoping you remember it. So how do we control the secretion of enzymes? So I've mentioned the enzymes. We'll come back to their chemistry, what they actually do. But first, let's talk about how we control them. It was cold in here, and now it's getting warm. So the trigger for secretion of enzymes by the exocrine pancreas, the trigger is fatty acids and amino acids in the duodenum. So there are cells there that detect those acids. Fatty acids, amino acids, fatty acids from fat digestion, amino acids from protein digestion. Those are detected when they enter the duodenum from the stomach. Remember what broke down peptides, what broke down proteins into some peptides or amino acids in the stomach anyway, was pepsin. So when chyme that contains these things enters the duodenum, it is detected in the small intestine, and endocrine cells in the small intestine secrete a hormone called cholecystokinin. I'll write that out. We'll just continue to use the abbreviation. Cholecystokinin. I think it's kinin. CCK. Cholecystokinin. That's the name of the hormone that the endocrine cells that some endocrine cells in the small intestine secrete. Like all hormones, it goes into the bloodstream, and the cells that respond to cholecystokinin are the exocrine cells of the pancreas, the acinar cells at the dead end of the pancreas. These guys. They have CCK receptors. We'll talk about, you can imagine what the slide is coming up. So CCK increases. CCK interacts with receptors to cause the exocytosis and secretion of those enzymes that were packaged into those secretory vesicles. So the enzymes go into the small intestine and digest fats, proteins, and starch, carbohydrate. So the trigger is chyme, specifically fatty acids and amino acids. The result is enzymes dumped into the small intestine for continuing chemical digestion of chyme. So here's the slide that you knew was coming. This is, so what cell are we looking at here? We're looking at a pancreatic acinar cell. This is a cartoon of this cell. This is the cell, this is the cartoon of it. So here are those vesicles all lined up, ready to go. Here is the CCK receptor, GQ coupled, calcium, you're off and running. CCK, there's the calcium story, calcium activating PKC, but calcium also directly triggering exocytosis. You've seen this story before. Calcium triggering exocytosis, calcium activating an enzyme that triggers exocytosis. So there's the story. This one's pretty easy. Now you could be wondering about all these other, like acetylcholine does this too. Yeah, it's true. Part of the, remember we have the parasympathetic nervous system, and the parasympathetic nervous system activates the cephalic phase of digestion. And part of the cephalic phase of digestion is to get the pancreas going, just like it was to get the stomach going. And that's that story. But the key thing here, the most important thing, is CCK. CCK is the most important stimulus for secretion of enzymes into the lumen of the pancreas, which then flows into the small intestine. We'll talk about secretin in a minute. Secretin is another hormone, and it's coupled to GS. So we're going to come back to that. So CCK is coupled to GQ, and secretin is coupled to GS. You've seen this story before, sort of, well, in the kidney. You had some GQ coupled receptors and some GS coupled receptors, and they were important for control of water and solute reabsorption. By the way, I am grading your exams. I am not done. I probably won't be done by the end of the day today, so I won't be able to give them back to you in lab. I hope to have them done by the end of the week, but there's also a self-phys exam. So when they're graded, I will put your scores up on. So if it helps relieve your stress over break to see your score, go to Moodle and see if it's there. If it won't be helpful for you to have a break, just don't go to Moodle. Okay, that little break. Let's talk about secretin. So the trigger for secretin is acid. When acid gets into the small intestine, any acid, gastric acid, fatty acid, amino acid, but mostly here we're talking about gastric acid. It's the most acidic acid we have. When acid from the stomach gets into the small intestine, it triggers other endocrine cells, not the ones that secrete CCK, but the ones that secrete secretin into the bloodstream, goes everywhere. Secretin activates the duct cells Secretin primarily activates the duct cells to secrete bicarbonate. But its secondary action is to augment the CCK-dependent secretion of enzymes. So secretin augments CCK-dependent enzyme secretion. And it triggers the duct cells, the pancreatic duct cells, to secrete bicarbonate to neutralize the acid that got there. Why do we need to neutralize acid? Well, the intestines are not adapted for digestion in an acidic environment the way the stomach is. So we evolved ways to neutralize the acid so that digestion could happen at a more neutral pH in the intestines, as opposed to the acidic pH of the stomach. Another one of these. Okay, let's see what you need to know from this one. I gave you this, I think it's on the next page. So what cell is this? So, remember, so this is a pancreatic acinar cell. This one is a pancreatic acinar cell whose job it is to secrete enzymes by exocytosis of enzyme-containing vesicles. This is a pancreatic duct cell whose job it is to secrete bicarbonate to neutralize the acids, the gastric acid in the stomach. How does this work? Well, so this includes both the mechanism for how it secretes bicarbonate and its control. So let's talk about the mechanism by which this cell, the duct cell, secretes bicarbonate. And you've already seen this in the kidney. Here's carbonic anhydrase making bicarbonate from CO2. CO2, is everywhere. There's carbonic anhydrase, there's CO2, and there's a transporter that you have already seen, a chloride bicarbonate exchanger that allows bicarbonate to exit into the lumen of the duct in exchange for chloride. So all of this other stuff, you don't really need to know. It's there. I mean, this figure was trying to be comprehensive. But the main story is CO2 gets converted into bicarbonate and is pumped out. That's the main story. Another story is that sodium bicarbonate cotransporter pumps bicarbonate in, and then the chloride bicarbonate exchanger pumps bicarbonate out. So those are the ways that we get pumping of bicarbonate. It's not exocytosis of bicarbonate, it's pumping of bicarbonate. Here's the duct. Here's the blood side, or the interstitial space outside the pancreatic duct. This is the basolateral membrane. This is the apical membrane. We're diffusing CO2 and then pumping bicarbonate, and then pumping bicarbonate from the body into the duct, where it is then secreted into the lumen of the digestive tract. The interesting thing is that secretin, remember secretin is the thing that activates bicarbonate secretion. And you'd say, okay, this thing should somehow be coupled to all of this, and it's not. Secretin, a GS-coupled receptor, GS, increases cyclic AMP, protein kinase A, all of the GS story goes on. But what this increases, what secretin increases, is a chloride channel. And by allowing chloride to flow out, it provides more chloride for pumping by exchange pumping bicarbonate out. So secretin indirectly activates this by providing the chloride that is necessary for this. Does it matter? Well, yeah, it matters because that's the molecular mechanism. You know, that's kind of what I'm about. So like, remember when we were back here, when we were back here, CCK was the main stimulus for enzyme secretion, but secretin was an augmenting stimulus for enzyme secretion. Secretin is the main stimulus for bicarbonate secretion of the duct cell, and CCK, where is it? CCK is around here somewhere. Well, it's not even shown, but you can decorate this. This is a CCK receptor as well as an ACH receptor. It augments bicarbonate secretion. So one is primary and the other is secondary. The primary for enzyme secretion is CCK, and the primary for bicarbonate secretion is secretin. The secondary for enzyme secretion is secretin, and the secondary for bicarbonate secretion is CCK. So there's a table in your textbook that walks through these effects. So we sort of walked through gastrin, and we got as far as this here. So let's go back just to sort of see what this table does for us. Gastrin is secreted by endocrine cells in the stomach that tell the parietal cells in the stomach to increase acid production in the stomach, stimulates acid secretion by the stomach. That's the main thing. So let's underline. Did I give you this one? Yeah. Let's underline the most important things here. Gastrin is produced by the stomach. Its stimuli are amino acids in the stomach. And what it does is it stimulates parietal cells to release acid. That's what gastrin does. CCK is secreted by endocrine cells in the small intestine. Those cells are stimulated by amino acids and fatty acids in the intestine that got there by digestion, from the stomach. And the most important thing here is it stimulates, right there, it stimulates enzyme secretion of the pancreas. That's the most important one right there. So underscore or highlight that stimulant. That's the main job of CCK. It also potentiates or augments secretin's actions, which we'll get to in a minute. So that's the next most important function of CCK. So that's number 2. And it also, and another important function that we haven't gotten to yet, but I will tell you about right now, is it stimulates the contraction of the gallbladder. So when we get to the gallbladder, we're going to be talking about CCK stimulating the squeezing of the gallbladder. So here's secretin. It is also secreted by endocrine cells in the small intestine. Acid in the small intestine is what triggers it to be released. When it is released, it stimulates the pancreatic duct cells to secrete bicarbonate. So this is its most important function, and this is its second most important function, potentiating the CCK effect on the duct cells. Those are the ones I want you to know from this table. Is that clear? Did you highlight or underline the ones that I want you to? Yes?
So the peptides in the stomach and parasympathetic nerves for gastrin is not Because
Right.
you only said amino acids.
I think amino acids and oligopeptides in the stomach are the most important trigger for secretion of gastrin.
Stimulates mitochondrial secretion.
Other questions about what to know and what to ignore on a complicated table like this? I'm not saying that things to ignore are wrong. I'm just saying that we have,
Second.
there's things to know and there's things to know that you don't know. The things to know later. Okay, let's do a couple of clickers. See how well this information is sunk in. Which of the following hormone combinations would maximally stimulate bicarbonate and enzyme secretion from the pancreas? Right, good, right. B is the right answer. CCK and secrete. Gastrin, anything that has gastrin in it, gastrin tends to, when it doesn't do anything to those things here. Gastrin doesn't help. Which of the following hormone combinations would maximally stimulate gastric acid secretion? Think you're getting it right? Oops. Gastrin alone. That's not a combination, so this was a trick question. But gastrin alone would maximally stimulate gastric acid secretion because, in fact, anything that had CCK in it tends to inhibit gastric acid secretion. So here's an interesting thing that once the chyme gets into the intestine, the endocrine cells of the intestine tend to help shut off gastric secretion, gastric acid secretion. It's a negative feedback. Okay, so here, match, this is a matching type of clicker. Here are the cell types: stomach endocrine cells, stomach parietal cells, small intestine endocrine cells, pancreatic duct, and acinar exocrine cells. Okay, those are the cell types. So let's go through them. Gastrin site of secretion and show me gastrin site of secretion. Let's do the first two. Show me gastrin. So I'm seeing a variety of answers here. Some of them are correct. Some of them are wrong. Gastrin's target. What is the target of gastrin? What cells does gastrin tell to do its thing? I should have broken this down one at a time. So gastrin's site of secretion is the endocrine cells, and gastrin's target is the parietal cells. Some of you put parietal cells as the secretion, the cells that secrete gastrin, and that's wrong. Endocrine cells secrete gastrin, parietal cells respond to gastrin. Okay, CCK site of secretion. Show me your answer there. Okay, I'm seeing a lot of correct answers, and CCK's target. Site of CCK secretion is the endocrine cells of the small intestine, and its target are the pancreatic cells, primarily the exocrine cells, but also the duct cells. And now you know the answer to the next two as well, same thing. Okay, so these are facts to put into your storytelling mode about how to tell the story of control of the pancreas. What cells, what it responds to, what cells respond by secreting the hormone, what cells respond to the hormone, and what that response is. Okay, CCK's target. Okay, this one, yeah, let's do this one. This breaks it down. What is CCK's target? This is breaking down duct cells as opposed to exocrine cells, endocrine cells, and gallbladder cells. I haven't stressed the gallbladder. What is CCK's target? I'll give you a hint. 2 correct answers. CCK target is B and D. CCK stimulates the acinar cells to secrete enzymes, the endocrine cells, exocrine cells, and it stimulates the contraction of the gallbladder. Secretin's target one best answer. Secretin's target are the duct cells of the pancreas. Primary target, its primary target. Its secondary target is the exocrine cells of the pancreas. Okay, let's do this some more, then we'll take a break. A stimulus for secretion of gastrin is, so these are now stimuli for the secretion of these hormones. A stimulus for the secretion of gastrin is most of you got that. A stimulus for the secretion of CCK is B and C. And a stimulus for the secretion of secretin is one best answer. Choose one best answer. You could make an argument for all of them, that D is the single best answer. Okay, let's take a break, and then we'll come back to what those enzymes do. Yeah.
Do you want us to know the branches of how they are potentially in each other's?
Comment about the exam. I told you I'm going to grade it, that I'll post the exam scores to Moodle when I'm ready, when it's done. The answer key is already posted to Moodle. So if you want to see what I wrote in my answers, you don't have to, but it's available to you now. That's available to you now, and the scores will be available to you later. And like I said, you don't have to do this now. You can do this later. All right. So now, let's talk about what the pancreas does for digestion. We talked about the enzymes and the bicarbonate. We talked about the hormones that control the secretion of enzymes and bicarbonate. All that stuff, we've got that going. Now, let's talk about what those enzymes and bicarbonate does for us. So here is a cartoon of the duodenum. Here's a cartoon of the pancreatic duct. Here is a cartoon of pancreatic juice carrying enzymes and bicarbonate from the pancreas to the intestine. They are lined by a cell type called enterocytes. That's just These are the epithelial cells that line the duodenum. So what's here is bicarbonate and those zymogens, the precursors. So one of the precursors was trypsinogen. It's an ogen, it's a precursor to trypsin. Just like angiotensinogen was the precursor to angiotensin that renin and ACE helped us make. Trypsinogen is the precursor to trypsin, and there is a resident protease on the cells that line the duodenum called enterokinase. Now, I have no idea why it's named that. Maybe it's a kinase too, but it's also a protease, and it cuts trypsinogen into trypsin. Trypsin is a protease. So trypsin is the enzyme that cuts protein. So it's going to cut the protein that's in the lumen of the small intestine into smaller bits. So like pepsin, it's proteolizing oligopeptides into shorter oligopeptides, or proteins into shorter oligopeptides, and some oligopeptides into single amino acids. Its protease action is to cut protein. Trypsin cuts the ogen for another hormone, another enzyme called chymotrypsin. Well, this is chymotrypsinogen. Trypsin cuts chymotrypsinogen into chymotrypsin, another enzyme that also cuts protein. So trypsin, which was activated by enterokinase, activates chymotrypsin. And trypsin, activated by enterokinase, cuts procarboxypeptidase into carboxypeptidase. So why are some of them ogens and some of them are pro? I have no idea. I wasn't there when they were named. Why is enterokinase named like that? I have no idea. Why is secretin? Secretin is sounds like such a generic name. Yeah, that's what it is. I can apologize, but it's not my fault. But these are the names of the things that you have to know. So trypsinogen, chymotrypsinogen, and procarboxypeptidase are the zymogens that are released by the pancreas, and then they are activated in the duodenum by a chain of events that starts with enterokinase activating trypsin, trypsin activating chymotrypsin, and trypsin activating carboxypeptidase by proteolizing the ogens, the zymogen, into their functional form. So now we've got this soup here of active proteases and bicarbonates. The bicarbonates neutralizing the gastric acid that came down here from the pyloric sphincter, and the enzymes are proteolizing amino acids. Don't forget there's lipase, which is going to help us with fat digestion, and there is amylase that's going to help us with starch digestion. But this is where we get a lot of protein digestion going on. So we're going to switch gears and talk about liver now. Here's the pancreas. Here's that pancreatic duct dumping into the duodenum. That's what we've been talking about. Now we're going to switch gears and talk a little bit of, well, more than a little, talk a substantial amount about anatomy. Both the pancreas and both pancreatic and liver anatomy. So let's just talk some terms, anatomical terms. Pancreas, head of the pancreas, tail of the pancreas, that's helpful. Left lobe, right lobe of the liver. We'll see that later. So this is the pancreatic duct, and there's a main duct and the main duct does this sort of loop-de-loop, and the secondary duct, there's a secondary opening here, both of these are dumping pancreatic juice into the duodenum through an opening called the sphincter of Oddi. Oddi must have been some anatomist who discovered this sphincter thing, and they named it for him. So this is, so like all the other sphincters, they open and close. They're closed between meals. It is closed between meals because we're not trying to dump pancreatic juice into the intestine between meals. The sphincter of Oddi opens when there is chyme in the intestine and allows the pancreatic juice to flow. It also, so when this is closed and the pancreas is secreting these things, and it happens some, that secretory juice is backed up into the gallbladder. So here's the gallbladder tucked in behind, it's sort of tucked into the liver, underneath the liver. And we're going to talk about some of these ducts. So pancreatic, this is plumbing. You have to know the names of some of this plumbing. Pancreatic duct, common bile duct, from the liver and from the gallbladder into the small intestine, into the duodenum. Pancreatic duct, common bile duct. The name of this tube right here, I'm going to give it to you now, it'll be on the next slide as well, it's called the cystic duct. So here's the slide that shows it maybe a little clearer. Here's a cartoon of the gallbladder. Cystic duct joining in to part. It joins in with another duct called the hepatic duct. So cystic duct is here, common hepatic duct, joined together to make the bile duct, or the common bile duct. Common bile duct dumps into the duodenum. The common bile duct came from the common hepatic duct and the cystic duct, merging to form the common bile duct. This is just anatomy, just the names of things. Of course, this is just memorizing the names of things. The hepatic duct is formed from the merging of the right and the left hepatic duct. Well, some of these names are helpful. Kind of, sort of. So we're going into the liver now. Hepatic duct, we're going to get into liver mechanisms today and then the Tuesday after break. This is the plumbing to know. This and this. So let's play some of this, let's play the game. Let me start with that. Will you start us off? Pick a name of something and point to it. I mean, point to the dot that it represents.
Would this be the right lobe of the
Yes, that would be the right lobe of the liver. This is the right lobe of the liver. Remember, you're looking at somebody else's liver. So what's on the, just like with the heart, this, even though it's on, it looks like it's on the left, it's on the person's right.
the tail of the pancreas?
Yes, tail of the pancreas.
Would that be right and left hepatic ducts?
Yes, right and left hepatic ducts. Right and left hepatic ducts.
And right below it's the common hepatic duct.
Common hepatic duct is right there. Yes, indeed. Oops, there. Common hepatic duct.
Duodenum.
That is the duodenum. Duodenum.
This one, the accessory pancreatic duct.
Okay, accessory. Yeah, somewhere, so this thing here is the so this is, see this loop going, that, yes, you were right. This thing here is the accessory pancreatic duct.
Head of pancreas.
Head of the pancreas is, yeah, sure, this thing right here. This whole thing, this dot right there is labeling the head of the pancreas.
Circular folds.
Circular folds are the folds inside the duodenum. Okay, I think we've done everything except for the so-called hepatopancreatic ampulla. Is that the only one left? That, in the previous slides, we called that the sphincter of Oddi. Just a different name. Well, the sphincter is the sphincter, the ampulla is this chamber. So, trying to illustrate. So this is the sphincter, and this sort of joining together is the ampulla, the hepatopancreatic ampulla. But I will not ask you to differentiate that. This you can call, if you point to this and you say sphincter of Oddi, I'm good. If you point to this and say hepatopancreatic ampulla, I'm good. But strictly speaking, the sphincter is the thing that closes the ampulla.
So are you gonna consider it redundant if we label like both? Like if you ask for like 13 like labels and
I
like
won't.
we're running out of label.
No, that'll count. Okay, we've done this one. Okay, who's got the laser? Yeah, okay. So we're gonna do this is the next one. It looks the same, but it's got different labels. I think if we were to play the game, this is the neck of the gallbladder, and this is the cystic duct, but obviously they're close. Head, body, tail of the gallbladder is not nomenclature I want you to know. So this is the neck of the gallbladder, but we don't care. This is the cystic duct, and we do care. At least I do.
This is the pancreatic duct.
Yep. This is the pancreatic duct.
This here is the hepatopancreatic sphincter.
Hepatopancreatic sphincter, another name for the sphincter of Oddi. This is the jejunum, following the duodenum.
And the body of the pancreas.
Yep, that's the body of the pancreas. So tail, head, body of the pancreas.
Sphinx.
Yeah, we did that one already. That is the hepatopancreatic sphincter. Let's see what's left. What is this? The bile duct. That is the bile duct or the common bile duct. Okay, maybe that's it. So papilla, so let's not worry about papillas. So this is a minor and this is a major papilla. This is the sphincter, whatever. I mean, anatomists can go nuts. But I think we've already labeled the ones that I want you to be able to identify and label. Okay? So now it's going to get really hard. This is the back of the liver. I didn't print this out for you because you don't need to know this. But we're going to see the back of the liver when we are with the cadaver this afternoon. So let's go through some of these things. So this is looking at the liver from the back. So the liver has been turned around, right? You're looking at the back of the liver. So this is now the left, and this is the right. Right? This is the right side of the liver, and this is the left side of the liver, because we're looking at it from the back. Okay. You might remember that the inferior vena cava was sort of went right, kind of not through, but next to, was adjoining to the back, the posterior element of the liver. So that's what we're seeing here. This is the left lobe, and this is the right lobe. And there is, this thing here is called the caudate lobe. Again, this is just me showing off. This is just so you've heard these words, but I will not test you on it. Caudate lobe, and this is the quadrate lobe. Here's the gallbladder, which is a structure that I do want you to be able to identify. We're going to talk about the veins. So let me go through this. Inferior vena cava, we did this. Caudate lobe is this bit right here. Right lobe is this bit over here. Bare area is this thing here. I don't have no clue why they bothered labeling that. Hepatic vein. We're going to talk about the blood flow to the liver after break. So the hepatic artery and hepatic vein are going to come at you after break. Gallbladder, we talked about. Common hepatic duct. Well, some tube in here, I can't even see it. Ligament, so this ligament here is called the falciform ligament. That's something you could label if you remember. Quadrate lobe we did down here. Hepatic artery, again, that's blood flow. So what's going to be interesting here about blood flow is we're going to have a hepatic artery and a portal vein, both of which are bringing blood to the liver. That's going to be an interesting twist. And then the left lobe over here. Okay, so the liver anatomy gets complicated on the reverse side. Let's talk about the functions of the liver. And the ones that you don't need to know. Okay? So we're going to end this class with these functions, and then we'll pick this up again after a break, which I hope has not caused you to forget everything. So the liver is important for the digestion of fat because it produces bile. So it produces bile, and it helps the pancreas produce the bicarbonate. It adds to the bicarbonate that the pancreas produces to help neutralize the gastric acid. So digestion of fat and neutralization of gastric acid are one of the important functions of the liver. You already know that the liver secretes angiotensinogen that you learned about for the renal section. You heard about albumin in the renal section, the protein that is in blood. Remember we talked about how albumin is not filtered at the glomerulus? Albumin is made by the liver. The liver is going to be a place where glucose is stored. So it releases glucose, and it stores glucose
when you've had a meal, and it releases glucose when you're hungry in order to maintain homeostasis of blood glucose. What else do you need to know? So, storing glucose, releasing glucose, storing fats, storing glucose as glycogen, a starch, storing glucose or releasing glucose from glycogen, storing proteins into fat. Another way to store calories between meals. Urea comes from the liver, and cholesterol comes from the liver. So those drugs, some of your parents or grandparents are probably taking statins to control their serum cholesterol. Those statins are acting on the liver and its ability to produce cholesterol. So do you need to know all 8 of these? I might ask you a question, say, name 5 important functions of the liver, or name 3 important functions of the liver, depending on how generous I'm feeling. So I would not ask you to name all 8, but I might ask you to name, more than half of them. Okay, so knowing at least some of these important functions of the liver. So after break, we will get to more liver and intestine anatomy.